Hello, I'm Vladislav Sandler, co-founder and CEO of Hemogenyx, and this is the second video from a series of informal videos that describe what we do, how we do it, and who stands behind our breakthrough research.
One of our main ventures at Hemogenyx is to develop novel models of humanized mice. These mice are immunodeficient mice, which we transplant with various combinations of hematopoietic cells, hematopoietic meaning blood forming. Therefore, when we inject these cells into mice, the mice will develop a human immune system. The sources of these cells vary from human umbilical cord blood to adult bone marrow, but our most recent model utilizes adult peripheral blood mononuclear cells. Previous models utilizing adult peripheral blood have the disadvantage of short lifespans due to rapidly developing GVHD, which is graft versus host disease, where the human graft attacks the host mouse, leading to the death of the animals within 4 to 6 weeks. And this short time period doesn't allow for any valuable experiments or data collection to be performed. At Hemogenyx, we've developed a proprietary protocol for processing peripheral blood cells that allow us to generate humanized mice that live over 100 days without developing GVHD. These mice have high levels of human chimerism in the peripheral blood, spleen, and bone marrow. These mice also generate human antibodies, both IgG and IgM. And the repertoire of these antibodies at least partially reflects the repertoire of the original human donors. We see a lot of potential for different uses of these mice, including testing of immunogenicity of novel biologics, rapid generation of human antibodies against human-specific pathogens, for the modeling of autoimmune diseases such as lupus, where there is aberrant antibody production and we can test treatments for lupus directly in these mice. Hemogenyx is currently working with Johnson & Johnson on further developing the mouse model of lupus. Also for testing treatments that modulate the human immune system, such as T-cell engaging bispecific antibodies like Hemogenyx's CDX antibody, or treatments that modify the existing immune cells like CAR-T or CAR-NK. We have many ongoing experiments utilizing these novel mice, and we hope to update you with the results soon.
Hi, I'm Elena Shrestha. I've been a scientist at Hemogenyx for a little over 2 years. Here, my work is basically focused on developing therapies for acute myeloid leukemia. At Hemogenyx, currently we have 2 pipelines underway for the treatment of AML. Our first approach to eliminate the cancer cells is to use bispecific antibodies that are highly specific. As the name bispecific suggests, these antibodies can bind simultaneously to 2 different antigens. On one end, these bispecific antibodies can bind to an antigen target on AML cells, whereas on the other end, they can bind to a receptor that's expressed on T cells, and these cells are immune effector cells. So the engagement of these T cells with the leukemic cells via bispecific antibodies leads to activation of T cells, causing them to release cytotoxic cytolytic agents into cancer cells, rendering them apoptotic and eventually leading to cancer cell death and elimination. In order to characterize these antibodies for their potency and efficacy, with the help of our team, I have established a number of in vitro assays, and we've also developed a robust AML mouse model that basically carries human cells, human immune cells that are functional. And apart from this, more recently I've been trying to develop an adoptive T-cell therapy by designing chimeric antigen receptor T cells against AML. In this approach, we are utilizing patient-derived T cells, isolated T cells are modified genetically to express chimeric antigen receptors, expanded ex vivo prior to infusing them back into the patient to treat their AML. The chimeric receptor on the surface of the T cells will then bind to the antigen that are expressed on AML, activating them and switching on their effector function to kill the cancer cells and hence eliminating them. And more recently, we've been trying different approaches to develop a safer CAR T because CAR Ts are known to have harmful side effects in patients that are undergoing clinical trials, and we're really trying to significantly reduce these side effects by creating a safer CAR T treatment.
Hi everyone, my name is Ronan and I'm a scientist. I would like to describe a few of the projects that we're working on at Hemogenyx. We're aiming to develop novel treatments for autoimmune diseases, including systemic lupus erythematosus, SLE. To do so, we're working in parallel on two approaches. One is a cellular model, and the other is a mouse model. To do so, we obtained blood samples from both SLE patients and healthy volunteers. Using 10x genomics technology for single-cell sequencing, we detected an abnormal subpopulation in SLE that led to the discovery of new targets that could be used for the development of new treatments. Additionally, in order to generate a mouse model, we took advantage of the humanized mice that were previously developed by Hemogenyx, and cells that were obtained from patients were engrafted into the mice, and eventually the mice developed key features of SLE, including production of autoreactive antibodies. To emphasize another very strong potential of Hemogenyx's humanized mice, I would like to refer to what is very relevant on the news today, the coronavirus outbreak. As we all became aware recently, pandemics are right around the corner, and while frontline medicine fails in treatment and prevention of the disease, we think that our humanized mice at Hemogenyx give us a significant advantage in the development of new treatments. Basically, blood samples that could be obtained from convalescent patients that fully recovered from the disease carry B-cell clones that produce neutralizing antibodies against the virus. Those clones could be engrafted in our humanized mice and eventually lead to the development of neutralizing antibodies as a treatment against the virus or an outbreak. I just hope that for the next time, we will all be prepared. Thank you.